Okay, hello everyone. Again, apologies about the technical issues. They seemed to be pretty complicated, but then in the end they got sorted out towards the end quite quickly, faster than we had feared. So that's great. Good morning, afternoon, evening, wherever you are in the world. Thank you for joining us. It's another episode of How Does the Brain Develop? And our hero today is Songhai Shi, who is, of course, needs little introduction. He's done fantastic work on all aspects of cortical development and brain development more generally. So just a few words of introduction. So Songhai did his bachelor at Tsinghua University in Beijing. He then moved to the Cold Spring Harbor Lab in the US to do a PhD with Robert Malinois, where he then moved to UCSF to do a postdoc with Yunang and Lily Jan. And in 2006, got his first faculty position at Sloan Kettering in New York, and at the same time at the Weill Cornell Graduate School. He was at Sloan Kettering and Cornell until last year. He, of course, became an associate professor, then a full professor. And since 2019, Songhai is a professor at the School of Life Sciences and the McGovern Institute for Brain Research at Tsinghua University in Beijing, China, and which I had the honor of visiting a few years ago. And it's just an absolutely fantastic place with amazing people doing great work. If you ever have a chance to go after this epidemic, don't hesitate. Songhai's work has attracted a lot of attention, a lot of awards over his career. I'll just mention a few: the McKnight Scholar Award, the HHMI Faculty Scholar Award, and so on. He has published on numerous topics his lab has published on numerous topics that deal with different aspects of brain development, from neural progenitor cells and their behavior, cell migration, of interneuron, for example, and the way they distribute in the cortex, neural circuit development, glutamate receptor regulation, different aspects of how the brain gets put together and how it works. And it's a real, I'll just won't take much more time. It's a real pleasure to have you, Songhai. I hope we can have another dinner in Paris soon, like a couple of years ago. That was wonderful. And looking forward to listening to you and hopefully meeting you again
Sure.
in the future. So please share your screen, and the floor, so to speak, is yours.
Sure. Thank you very much, Bethan. Let me see in terms of sharing the screen. Just a second. Give me a second. Let me see how to get the screen being shared. Yeah.
Sorry about that, guys. It worked just a minute ago, so hopefully there isn't a... So if you just toggle the mouse on your video, you should have the share screen.
Okay.
There we go.
Great.
Perfect. Okay.
Thank you very much, Bethan, for the kind introduction. It's great seeing you, and also like to thank Danny and Bethan, all the organizers, for the opportunity to speak here. So it's great to have this venue to stay connected during this special time. And also I see old friends, young Christoph is also here. So I'd like to take this opportunity to share with you our effort trying to understand the assembling of the neocortex. Can you hear me okay? So the neocortex is one of the most complex brain structures, and it's essential for all high-order brain function, from perception to language to cognition. So one of the clear indication of its complexity is its content of a large number of diverse neurons and glial cells. So in human neocortex, we have about 86 billion of neurons and also about 100 billion of glial cells. So in a simplistic view, the research in the field is trying to understand two important questions. One is, how do we generate this large sets of diverse neurons and glial cells during the development? Essentially, the questions of the neurogenesis and gliogenesis. The second important line of question is, how are those neurons and glial cells assembled into functional circuit to operate to support all those high-order brain function? So the outline of today's presentation, I hope to cover both aspects. The first part is about histogenesis of the mouse neocortex. Essentially, we want to figure out how the progenitor cell behaves during the cortical development and what's the process of neurogenesis and gliogenesis. If we have time, I also like to touch upon how this early development process of neurogenesis and gliogenesis may impact the functional circuit assembling and the map formation in the neocortex. So we all know that neocortex is a highly organized brain structure. So one of the most prominent organization of the neocortex is the horizontal laminar structure, as initially discovered by Romani and Cahar by joining the neocortex based on his observation of the brain sections. This is a human neocortex that draw from the top of the brain to inner white matter. We can see there is this very nice organization of neurons into horizontal layers. So this 6 layer of horizontally organized cells is one of the hallmark of the neocortex. Whenever you see this type of 6-layer laminar structure, we know we are in the neocortex. Of course, one of the fundamental questions is, how are those numerous and diverse neurons and glia cells generate and organize into those horizontal layers during the development? So research done in past several decades has gained quite a bit of insight into this process. For example, we take the mouse brain as example. This is embryonic day 13 mouse brain that we do a Clonal section. This is what we see. We first see the ventricle. That's a natural ventricle where the cerebral fluid floats through in this space. Then we have a zone called ventricular zone. That's where the stem progenitor cell reside. And those stem progenitor cells will actively divide in the ventricular zone and produce neurons that will radially migrate to occupy a structure called the cortical plate. That's the future neocortex. So as the development proceeds, the progenitor cells in the ventricular zone will produce a large number of neurons that will occupy and expand the cortical plate that become the future neocortex. So if we zoom in, we can roughly divide this early phase of neocortical development into 3 stages. The first stage is the proliferation of the neural stem cells, initially neuroepithelial cells, to give rise to radioglial cells. This very specialized bipolar type of glial cells. And those cells initially thought to be the supporting cells of the neocortex, but later on they found those cells are also the progenitor cells, stem cells in the developing neocortex. So initially they will symmetrically divide to amplify, to have a sufficient pool of the progenitor cell to support the later neurogenesis and gliogenesis. Then at some point, those proliferating neural progenitor cells will enter a phase called neurogenesis phase, which means they will start to produce neurons. There are 2 important features of the progenitor cell during this neurogenic phase. One is that they switch from symmetric proliferative division to asymmetric neurogenic division, which means they will divide to renew itself at the same time to produce a neuron, or an intermediate progenitor cell. They will go on to divide additional round to produce more neurons. And also an important feature for the laminar formation is that the neurons that are born at the similar time, they will migrate as a cohort to occupy a similar laminar depths in the developing neocortex. So we have this birth state dependent inside out lamination of the neocortex. So it's very much like we construct a skyscraper building. We build the neocortex by stacking the layer of neurons on top of each other. So early born neurons being the deep layers of the neocortex, and later born neurons sitting on top of it beyond the superficial layer of the neocortex. And this inside-out lamina formation clearly is an important basis for the horizontal layer formation of the neocortex, but also it has important functional implication. Not only the neurons born at different time that occupy different lamina of the neocortex, but their function also quite different. For example, early born deep layer neurons are largely those neurons will project outside of the neocortex to the subcortical structures such as the spinal cord, cerebellum, or thalamus. But most of the superficial layer neurons will be those neurons that project within the cortex, either apsolateral or contralateral, to mediate cortical-cortical interaction. And at certain stage, at the completion of the neurogenesis, then some of those progenitor cells will go on to produce glial cells, largely those macroglia, the astrocyte and oligodendrocyte. So we can roughly divide this early process of cortical development into these three phases. But one of the fundamental questions is that how do individual radioglial progenitor cells divide to produce diverse neuron and glia? Because we know there's at least based on current understanding, there's 80 to 100 different types of neurons and even more types of glia as well. So we hope to understand how do individual radioglial progenitor cells divide to produce these different types of neurons and a different number of neurons and glial cells. So we hope to do a lineage reconstruction of individual dividing radioglial progenitor cells to have a quantitative understanding of the construction of the neocortex. To achieve that, we took advantage of rather sophisticated clonal labeling technology that developed by Li Chen Lab based on initial fly Markham system. So in mammalian, it calls the mosaic analysis with double marker called MADAM. So the basic idea is that you put a split GFP and a split IFP into the same chromosome locus. So if this happens in a dividing progenitor cells, as a progenitor cell divide, you go through the chromosome replication, and also you can trigger Cre-dependent interchromosome recombination, which is a relatively rare event compared to the intrachromosome recombination. So you can reconstitute the GFP and the IFP. And as the progenitor cell divide to produce two daughter cells, depending on chromosome segregation manner, if it's X segregation, those reconstituted GFP and IFP will be inherited by two future daughter cells. So this way you can permanently enable two initially generated daughter cells with two distinct color, GFP and IFP in this case. So this provides unprecedented resolution on the division mode and as well as the output of the dividing progenitor cell at the clonal level. And in fact, you can couple this type of labeling technology to both the spatial temporal controls to use Cre-ER, for example. We can use EMX1 Cre-ER to achieve the forebrain specificity. Then we can induce the recombination at the defined embryonic stage, then analyze the output of neurons at various postnatal stages. So for example, this is one of the experiment we done quite a few years back by using EMX1 Cre-ER, which enable cortical progenitor cells, and then do the induction at the E10 and then look at the brain at the P21. So we can do serious section to reconstruct the entire brain by recover all the labeled cells. As you can see, we can see a very nice clone of X segregation and also see a Z segregation with both GFP and TD tomato inherited by the same daughter cells. So this type of analysis can recover the output and also spatial organization of the entire neural progeny of the dividing progenitor cells, for example, at E10. So if we do this systematically, we found that the behavior of the radioglial progenitor cells in the developing mouse neocortex is highly organized. For example, before E11 and E12, they mostly go through the proliferative symmetric division, and also their division is highly orchestrated in a sense that every round of division, the radioglial progenitor cell will lose their proliferative potential. So as they go through more division, they will progress towards differentiation. At around E11, E12, they will switch from a proliferative division to neogenic asymmetric division. And once they enter the asymmetric neogenic division, individual radioglial progenitor cells produce about 8 to 9 neurons. We think this is a unitary output. And then towards the end of the neogenesis, about 15 to 20% of those radioglial progenitor cells will go on to produce glial cells, to produce both astrocyte and oligodendrocyte. So we think the behavior of the radioglial progenitor cell, at least in terms of number of neurons it produces, is highly organized. It's not a stochastic or random process. But of course, some of you more familiar with the area that we know that a recent paper published by Oscar Marin's group suggesting there is a stochastic random process of neogenesis. So I'd like to spend a few minutes just to discuss those two different views of the radioglial progenitor cell mediate neogenesis in the neocortex. So the basic idea is that based on this Madam labeling, if we look at purely on the size, which means the number of neurons individual neogenic radioglial progenitor cells produce with this type of histogram, or the distribution of the clone size, which can be approximated by a normal distribution, indicating there is a relatively defined output about 8 to 9 neurons per dividing radioglial progenitor cells. And also we found that those 8 to 9 neurons that will occupy both the superficial and deep layers. In fact, we think that this reflects a rather coherent radioglial mediate neogenesis model, in a sense that the radioglial progenitor cell is largely homogeneous in a sense to produce deep layer neurons first, then progress to the superficial layer neuron. But this doesn't mean every radioglial progenitor cell will produce the same laminar configuration of the output. So there will be variability in terms of the laminar occupation. One of the important indications of this coherent model is that we find there is an inverse correlation between early born deep layer neurons versus late born superficial neurons by individual radioglial progenitor cells. As shown here, if you look at the individual clones, the number of superficial layer neurons versus the number of deep layer neurons is actually inversely correlated, indicating the output of individual radioglial progenitor cells is likely to be a defined number, that if you produce more deep layer neurons, you may produce fewer superficial layer neurons. So we think that individual radioglial progenitor cell by large has a rather coherent output by producing about 8 to 9 neurons. But there is a variation in terms of the individual radioglial progenitor cell enters neurogenesis and exits neurogenesis, which means that not all the radioglial progenitor cells are synchronized in terms of their neurogenesis program. And this may create variation in terms of the size and also laminar distribution of the output of neurons. So in fact, with a similar type of approach, even in Oscar's paper, they see a rather similar behavior. In other words, if they use the Madam approach, they find a rather relatively similar output of individual radioglial progenitor cells in terms of number of neurons that produce in the neocortex. However, they have another method of looking at the output of the radioglial progenitor by using the Cree-dependent reporter, GFP, coupled with EMX1 Cree-R. In this type of labeling, they found there is a population of the clone which has a relatively small number of neuronal output, and also can produce only superficial layer neurons or only deep layer neurons. And this, of course, they still debate whether that reflects a radioglial progenitor cell or some intermediate progenitor cell output. This is a debating issue. But nonetheless, based on this data, they propose this heterogeneous radioglial progenitor cell neogenesis model, which means that radioglial progenitor cell are inherently heterogeneous. Some will only produce deep layer neurons, and some will produce only superficial neurons, while majority produce both superficial and deep layer neurons. Another essence of this model is that the neogenesis at individual layers will be stochastic or close to the random. So in other words, they produce one layer of neurons doesn't influence the production of the neurons in another layers. So those two different models has different predictions in terms of the laminar output of the individual radioglial progenitor. For example, if this, based on this coherent radioglial progenitor cell model, we think this output is relatively defined. So you're shifting this program as you're moving through the developmental stage, for example, from E12.5 to E16.5. So you have this shifting normal distribution as development proceeds, and also this produce different neurons in different laminas as development proceeds. While in this model, because it's a stochastic and also random process, so in terms of generating the output, it goes rather complex modeling to predict its final output. Of course, there's no absolute proof of those two different models, and each model has its own prediction in terms of the output and laminar identity. So I think a lot more work needs to be done to really tell between those two different models. So I'd like to move from neogenesis to gliogenesis. We know that radioglial progenitor cell in the developing neocortex is capable of producing both astrocyte and oligodendrocyte. But one of the key questions is, at what stage do radioglial progenitor cells switch from neogenesis to gliogenesis? And when they make this switch, what is the quantitative behavior of the radioglial progenitor cell? In other words, if they can produce both astrocyte and oligodendrocyte, how many and what type of astrocyte oligodendrocyte produce? Just a rough, brief introduction of the cortical glial cells. So there are two major types of glial cells in the developing neocortex. One is the macroglia, the astrocyte and oligodendrocyte. Of course, there are also microglia. But actually, microglia has a different developmental origin. It comes from mesoderm, from yolk sac. So the dorsal radioglial progenitor cells, while they are producing neurons, as development proceeds, they're also capable of producing both oligodendrocyte and astrocyte. Of course, there is also ventral source of oligodendrocyte for the cortical oligodendrocyte based on the pioneer work done by Casares and Richardson. But we want to know when dorsal radioglial progenitor cells switch from neogenesis to the gliogenesis, and how and what type of glial cell do individual gliogenic radioglial cells produce. And to address this question, again, we took advantage of the MADAM approach. We decided to perform a systematic and quantitative analysis of gliogenesis by the dorsal radioglial progenitor cells using MADAM. So to do that, we set it up to label single neogenic radioglial progenitor cells at around E13 to E17 while they are producing neurons, because we want to catch the neogenic radioglial progenitor cell as they switch from neogenesis to the gliogenesis. This way, we can get the complete output of all the glial cells from individual gliogenic radioglial progenitor cells. And also, we focus on green-red G2X segregation clones. This way, we can know this is coming from a dividing progenitor cells. As I mentioned, there are only about 15 to 20% of neurogenic radioglial progenitor cells go on to produce glial cells. So this is actually a rather difficult task, because we have to connect lots of clones to recover enough of the clone with glial cells. So this is largely the work of two talented students at Tsinghua, Zhongfu Shen and Yang Ling, who carried out the task. So this is one of the examples. So after we do the induction, for example, at E13, then we look at the brain at P30, we perform the serial section to recover all the labeled cells. So this is a consecutive section of this brain in one hemisphere. So you can see both the green and red labeled cells. So those small cells, as shown by open arrow, those are neurons. They have fine processes. And also those bright cells, as shown here, those are glial cells. We can see both the astrocyte and oligodendrocyte, which you can, after you have enough experience, you can relatively easy to tell that based on their morphology. For example, this is a typical astrocyte morphology. This is a typical oligodendrocyte morphology with parallel fibers that are likely to be important for the myelination of the axons. And to further confirm their identity, we perform immunohistochemistry by using the well-characterized markers. For example, with neurons, we can use neuron. With astrocyte, we can use ALDH1L1. With oligodendrocyte, we can use OLIG2 that nicely match with their morphology of their identity. So then we perform a systematic analysis of all the glial-containing clones that labeled as the radioglial progenitor switch from neogenesis to gliogenesis, which means this clone will contain both neurons and glia. And this is a movie. It doesn't work. And also, during this experiment, we perform OLIG2 staining to separate the oligodendrocyte versus astrocyte. And this is a reconstruction of the clone. That the rounded dot represent the neuron, the soma of the neurons. And this is a typical asymmetric dividing clones that initially produce neurons that is in the deep layer. So this reflects the firstborn neurons by this asymmetric dividing radioglia progenitor cells. And all the red cells, including both neurons and glia, are coming from renewing radioglia progenitor cells go with multiple rounds of division. Initially produce all those red neurons, then will produce all those red glia cells, including oligodendrocyte and astrocyte. So the first thing we notice that if we look at the overall size of neurons and glia cells that enabled by radioglia progenitor cells at different embryonic state, you can see there is a clear difference. For example, we look at the average number of neurons in individual clone, you can see the size progressively reduced. This is consistent with the progressive reduction of neogenesis of individual radioglia progenitor cells as they shift from deep layers to superficial layers. In contrast, if you look at the average number of glia cells in individual clone, the number actually stays relatively constant. In other words, each individual gliogenic radioglia progenitor cell produces about 10 glia cells, including both astrocyte and oligodendrocyte. And this is consistent with the idea that the gliogenesis largely occurred at the completion of neogenesis. So no matter when you label the progenitor cells, the output of the glia cells is at the later stage. So the size or the number of glia cells stay constant. But of course, if you label the clone as they switch the gliogenesis, the size of the glia will change, become smaller. But prior to their switch to the gliogenesis, then all the output of the glia cell will stay the same. And also we observe 3 different types of the clones. The clone contain only astrocyte, as shown here. You produce red neurons and green radioglia progenitor cell produce green neurons and green astrocyte, or only contain oligodendrocyte in this type of oligodendrocyte only clone, or produce both astrocyte and oligodendrocyte, indicating there's 3 different ways of producing glia when radioglia progenitor cells switch from neogenesis to the gliogenesis. One of the things interesting is that if you look at the overall ratio between these 3 different types of clone, no matter when you label the progenitor cells, they stay more or less the same. On average, the astrocyte versus astrocyte plus oligodendrocyte versus oligodendrocyte clone is about 60 to 25 to 15%, indicating this is inherent feature of radioglia progenitor cells when they switch from neogenesis to gliogenesis. So come back to the question, can we ask, when do radioglia progenitor cells switch in vivo from neogenesis to the gliogenesis? So with the MADAM labeling, you actually can tell. The idea is that if the radioglia progenitor cell undergo the last round of neogenic division, which means they will produce last neuron, then it will switch to produce glia. So in this case, neuron will be in one color, the glia will be another color in the clone. So this way we can explicitly tell when the radioglia progenitor cell will switch from neogenic division to gliogenic division. So which we call those type of clone called transitioning clone. So the idea is that when the radioglia progenitor cell finish neogenesis, they will transition to gliogenesis. During this transition, the clone will have neuron in one color, as shown here, neuron in red, and glia cells in green. So all those are coming from a renewing radioglia progenitor cell that only produce glia cells. Of course, prior to the transition, you will see the clones with neurons in both color. This means that the labeling time of the radioglia progenitor cell is still in the process of producing more than one round of neurons. And also we can see after the transition, the clone only have the astrocyte glia cells. They actually have astrocyte oligodendrocyte in different colors. So with this type of labeling, we can tell during the mouse neocortical development, the majority of the radioglia progenitor cell make the transition from neogenesis to the gliogenesis around E15 to E16. By E17, most of the radioglia progenitor cell finished neogenesis and transition to gliogenesis. So the transition occur in a progressive way, but largely occur at E15, E16, and by E17 is largely complete. And also with the capability to identify the transitioning clone, we actually can find the transitioning clone also exist in the 3 different types. For example, they can be transitioning only produce astrocyte, or transitioning to produce only oligodendrocyte, or transitioning to produce both astrocyte and oligodendrocyte. And interestingly, regardless when the radioglial progenitor cell will make the transition, of course, at early stage, the transitioning radioglial progenitor cell is relatively few compared to later, but the overall ratio is similar, as shown here, and this ratio fits very well with the overall cloning type of the astrocyte plus oligodendrocyte versus oligodendrocyte with astrocyte only is about 60 to 25 to 15%. And this data strongly suggests that this capability of radioglial progenitor cell to produce glial cells and in these 3 different types of lineage progression is likely to be an intrinsic properties of radioglial progenitor cell, as no matter when they make the transition, the overall output stays comparable. Another interesting thing, we noticed that there is independence between the astrocyte production and oligodendrocyte production, as shown here, regardless whether a clone contains both astrocyte or astrocyte and oligodendrocyte. If you look at the astrocyte number, they're more or less the same. Similarly, regardless whether a clone contains the astrocyte, the number of oligodendrocyte, either in the pure oligodendrocyte clone or the oligodendrocyte plus astrocyte clone, the size stay more or less the same. If that's the case, you will predict if the clone contains both astrocyte and oligodendrocyte, the total number of glial cells will be similar to the summation of the astrocyte only clones or oligodendrocyte only clone. This is largely the case. The overall size of astrocyte plus oligodendrocyte clone will be close to the summation or the sum of the astrocyte only clone, oligodendrocyte clone. This indicating that the astrocyte production, oligodendrocyte production seems to not influence each other, and then likely because of early lineage separation between the astrocyte and oligodendrocyte production in the process. Another important feature we noticed that is that we find the astrocyte and oligodendrocyte comes in local clusters. For example, as shown here, if you look at the astrocyte or oligodendrocyte in the clone, it comes in small local clusters. So we call those subclusters. Actually, the similar observation also being found by a recent study by laboratory on the astrocyte production. And we find that in those individual subclusters is either astrocyte or oligodendrocyte. There is very little subcluster contain both. So we think that those individual subclusters likely reflect the output of the local intermediate glial precursor cells. So they will produce a similar type of glial cells, either astrocyte or oligodendrocyte. As shown here, these are typical astrocyte subclusters and oligodendrocyte subclusters. You can see within the subclusters, the astrocyte, oligodendrocyte, they actually often touch each other. This raises the interesting possibility they may reflect some of the kind of the glial cell domain. So whether it has any functional implication in the brain-blood barrier formation or interaction with local neurons is one of the interesting questions. But this also raises another interesting possibility, as I mentioned, that those individual subclusters may come from a single intermediate precursor cells that produce either astrocyte or oligodendrocyte. Can we test this? We know that there's those intermediate precursor cells that exist in the neonatal neocortex that are responsible to produce astrocyte or oligodendrocyte. So we decided to use nesting tree to identify and label those intermediate precursor cells and to see whether they can produce those local clusters and then generate those defined types of glial cells. So with nesting tree coupled with MADAM, actually we can label, for example, at P1, P3, P5 to identify those local astrocyte or oligodendrocyte clones as shown here. So for example, in this G2X nesting MADAM clone, we can see 2 astrocyte that forms a local clone as shown here, or in this case have 5 oligodendrocyte forms a local clone. That. So we think that there are fit-restricted intermediate glial precursor cells that exist in the neonatal cortex. They probably are produced by gliogenic radioglial progenitor cells. And also within those individual clones, you either only have astrocyte or oligodendrocyte, but there is no mix of astrocyte plus oligodendrocytes, consistent with the idea that intermediate glial precursor cells is fate restricted. So by looking into those subcluster or intermediate precursor clone labeled by nesting CRE-ER, we actually find in the astrocyte clone there is about 2 to 3 astrocytes on average. If you look at the histogram, it shows this peak distribution, indicating that individual intermediate glial precursor cells has a relative defined output of the astrocyte. For example, produce on average about 2 to 3 astrocytes. Similarly, with the oligodendrocyte, you also produce on average 4 to 6 oligodendrocytes, but also show a peak distribution, indicating that the intermediate glial precursor cells output is relatively defined, about 2 to 3 astrocytes or 4 to 6 oligodendrocytes. But what about the number of the intermediate precursor cell produced by gliogenic radioglial progenitor cell during the embryonic stage? To do that, we can analyze the number of subclusters within the EMX1 CRE-ER enabled clone. So this way we can tell how many subcluster, how many intermediate precursor cell it generates. For example, within individual gliogenic radioglial clone, we can see on average about 2 to 3 subclusters. But look at the subcluster distribution. It shows this geometric distribution, indicating that the output of intermediate glial precursor cells by gliogenic radioglial progenitor cell is largely stochastic. So it doesn't have a relatively defined output. Similarly, the production of intermediate oligodendrocyte precursor cells by individual gliogenic radioglial progenitor cell also shows a geometric distribution. So indicating that the production of the glial cells has both defined and also stochastic component, which is different from neogenesis. So the production of the number of the intermediate glial precursor cells is stochastic, but the number of the glial output by individual intermediate glial precursor cells is relatively defined. So we can consider those intermediate glial precursor cells like intermediate progenitor cell for neurogenesis. So overall, the design of the gliogenesis and neurogenesis start from radioglial cells divided to produce intermediate progenitor cells is not the same, but there are some difference between neurogenesis and gliogenesis. The main difference is that the number of the intermediate glial precursor cells appears to be stochastic, produced by gliogenic radioglial progenitor cells. So to summarize this part, we think that the dorsal radioglial progenitor cells is capable of producing both astrocyte and oligodendrocyte, as previously shown. And this segregation of the astrocyte and oligodendrocyte production occurs quite early in lineage. And also they go through producing the intermediate oligodendrocyte precursor cells or intermediate astrocyte precursor cells to produce astrocyte oligodendrocyte. The production of those intermediate astrocyte precursor cells or intermediate oligodendrocyte precursor cells by radioglial progenitor cell is stochastic, but individual intermediate astrocyte precursor cell produce about 2 to 3 astrocyte. Individual intermediate oligodendrocyte precursor cell produce about 4 to 6 oligodendrocyte. And they forms local clusters or local glial domain. So what about the subtype? We know that the emerging evidence suggesting that the glial cells in the developing in the neocortex is also diverse. They may also have lamina identity. But in this analysis, we took a very simplistic view. Based on previous literature, we grouped the astrocyte based on their location and GFAP expression. So for example, there are layer 1 astrocyte that express strongly GFAP, but also have this unique morphology of both bushing and thick branching in combination. Of course, we have protoplasmic astrocyte has fine processes, or the fibrous astrocyte that have thick processes that express high GFAP. So based on these 3 different subtypes of astrocyte, what we can find is that within individual subclusters, it's largely the pure subtype. For example, largely either to be protoplasmic astrocyte or fibrous astrocyte or layer 1 astrocyte with very little subcluster containing mixed subtypes, suggesting that individual intermediate astrocyte precursor cells largely produce the same subtype of astrocyte. But if you look at from gliogenic radioglial progenitor cell level, you can see majority of the radioglial progenitor cell produce a single subtype of astrocyte, but there is about 20 to 30% of radioglial cells produce more than one subtype. If you look at those with more than one subtype, they're either to be the Neocortical astrocyte plus protoplasmic astrocyte, or protoplasmic astrocyte plus fibrous astrocyte, but not the Neocortical astrocyte plus the fibrous astrocyte, indicating the output of individual radioglial progenitor in terms of subtype is also not a random process. If you look at with only a single subtype, of course, as expected, most of them are protoplasmic only clones, with some of them with fibrous astrocyte clone. Similar with oligodendrocyte, we know oligodendrocyte, at least besides the regional differences, within a similar region, it mostly reflects the mature state of the oligodendrocyte. It can either to be the oligodendrocyte precursor cells. I like to emphasize this is different from those intermediate oligodendrocyte precursor cells. Those are cells that can exist in the adult brain. And also the non-mature, non-myelinated oligodendrocyte or mature oligodendrocyte is shown here. They can be distinguished based on the morphology and marker expression as shown here. So if you look at the individual subcluster of oligodendrocyte from the radioglial progenitor cell clone, you can see most of them contain only one subtype with very little mixture of more than one subtype, in this case maturing state, indicating that individual intermediate oligodendrocyte precursor cells largely produce the same mature state of oligodendrocyte. And also, most of them has a single state. If you look at the radioglial progenitor cell clone, few of them contains more than one state of oligodendrocyte, either to be oligodendrocyte precursor cells with mature oligodendrocyte or non-mature oligodendrocyte plus mature oligodendrocyte. So this indicating that the individual intermediate precursor cells largely produce the same type of oligodendrocyte or astrocyte. So finally, I'd like to touch upon the potential molecular regulation of this process and its potential link to the brain tumor. We know that NF1, neurofibromin, is a very critical molecule for regulating the glioma formation. Basically, is RAS activity suppressor, and the mutation has been linked with glioma occurrence. So we can take advantage of the MADAM labeling by coupling the labeling with the mutation to perform mosaic clonal loss of function analysis. In other words, we can select a mutant or removal NF1 in one of the siblings of the dividing progenitor cells, where the other serve as a wild-type control. So for example, we can link the NF1 allele because they are on the same chromosome with this MADAM allele, the chromosome 11. So we can link the mutant NF1 allele with the red color. So in this individual clone, the red cells will be the mutant and the green cell will be the wild type. So for example, we can look at those gliogenic clone by comparing the green neurons, red neurons, green glial cells versus red glial cells to know whether NF1 mutation have an impact on neurogenesis or gliogenesis. For example, as shown here, you may notice that if you compare red glial cells versus the green glial cells, you actually can see the red green glial cells in individual clone has more in terms of the size compared to green neurons in individual clones. But if you look at the neurons, there is no obvious difference. So if you quantify, you can see the F1 deletion does not affect neurogenesis, but it can cause a drastic increase in gliogenesis. If you look at the average number of glial cells in the F1 mutant red lineages, you can see there are not more glial cells. One of the interesting things is that even the F1 deletion can increase both astrocyte production or oligodendrocyte production, but the extent of the fold of the increase is quite different. For the astrocyte production, the increase is about 2 to 3-fold, but for oligodendrocyte production, we have about 10 to 15-fold of increase. So if you look at histogram or the distribution of the size of the clone, you can see that the astrocyte oligodendrocyte clone clearly all shift towards the large size. But if you look at the oligodendrocyte, you can see those huge clones which more than 10 standard deviation away, indicating that NF1 even deletion can infect both astrocyte and oligodendrocyte production, but they can affect oligodendrocyte production much more dramatically. This is as reflected. We can often see there's a huge oligodendrocyte clone with thousands of oligodendrocyte in individual clone. And also, interestingly, all those cells in those large clones are oligodendrocyte precursor cells that express NG2. So those results suggest that this oligodendrocyte production is likely more susceptible for tumor formation because it can have much dramatic proliferation capacity and administered because of the NF1 deletion. So what is a clear difference between the regulation of NF1 or astrocyte or oligodendrocyte production? We actually go through this quantitative analysis, found that the NF1 deletion can increase both the number of the intermediate astrocyte precursor cells or intermediate oligodendrocyte precursor cells, which means individual gliogenic radioglial progenitor can produce more intermediate glial precursor cells for both astrocyte or oligodendrocyte. However, the NF1 deletion does not affect the output of individual intermediate astrocyte precursor cells. They produce about 2 to 3 astrocytes. But NF1 deletion can dramatically increase the output of individual intermediate oligodendrocyte precursor cells. Some of those can produce hundreds of oligodendrocyte precursor cells, and this is likely to be the reason for the huge clone that we observe. And this type of process may relate to the tumor formation because of the NF1 mutation. So to put this part together, we think that with this type of systematic quantitative analysis, we can get a quantitative understanding of the gliogenesis by dorsal radioglial progenitor cells, and that goes through this rather organized process to produce astrocyte and oligodendrocyte, and this may also have a relationship with brain tumor formation. So this is what we think the histogenesis program of the mouse neocortex based on this type of analysis. We think the behavior of the radioglial progenitor cell at each stage of proliferation, neogenesis, and gliogenesis, they have a rather defined behavior from the round of proliferative division it can go through, from a number of neurons it can produce, and the relative occupation in the superficial versus deep layer of the cortex, and also the number of the astrocyte or oligodendrocyte it produces and what type of process it goes through. And I'd like to mention that some of those radioglial progenitors towards the end also produce ependymal cells, adult neural stem cells, or olfactory bulb interneurons. So even those are minority of the output, but they also produce those important cell types. So we also in the process try to understand what is the overall quantitative behavior of generate this type of cells in the developing neocortex, and also, as well as in the olfactory bulb. So I still probably have about 10 minutes. I will quickly introduce the second part in terms of one of the main reasons we try to understand the histogenesis of the neocortex in terms of neogenesis and gliogenesis is to try to understand how we can assemble a functional neocortex. In other words, what's the influence of the early process of neurogenesis and gliogenesis can influence the circuit assembling and the organization, a functional organization of the neocortex. So we know that the functional organization of the neocortex is largely influenced by the early work of Hubert Wiesel and Moncasso, the basic idea of the functional column. So orthogonal to the horizontal layers, we have those functional circuits in a vertical direction that we call functional column, basically a group of neurons spanning from the pure surface to inner white matter can wire together to form synapse specifically and to have a defined physiological function, for example, in the visual cortex orientation tuning columns. In fact, Hubert Wiesel made the suggestion that this type of functional column can be uniform and repetitive in the sensory cortex, for example, in the visual cortex, and also in the somatosensory cortex and the auditory cortex. So, of course, one of the fundamental questions is that how are those relatively uniform, repetitive clonomic circuit being assembled in the neocortex. In other words, what would be the mechanism to drive this repetitive assembling of the vertical circuit in the neocortex? Of course, we can come back to the early process of cortical development, because we know that the neurogenesis and neural migration is largely occurred in the vertical direction. So could those early process of neurogenesis and neural migration influence the functional circuit assembly in the neocortex? So over the past 10, 15 years, and we have been looking into this systematically, we think that the early process of neurogenesis and neural migration do influence functional circuit assembly. In other words, the sister excitotory neurons come from same neurogenic radioglial progenitor cells. They migrate along their mother radioglial fiber to form a developmental column, and this developmental column can assemble into a functional circuit. For example, we know that individual radioglial progenitor cell can produce a unitary output of deep layer and superficial layer neurons. And those lineage-related sister neurons can go through this bursted dependent inside radial migration along the mother radioglial fiber, and this will ensure the lineage-related sister excitotory neurons to encounter each other to support the preferential formation of the gap junction or electric coupling. And this electric coupling can promote activity synchronization between the sister excitotory neurons. This can promote preferential chemical synapse between sister excitotory neurons, and this can drive the functional clonomic circuit formation with a defined physiological function. For example, they share similar orientation tuning synactivity. Of course, this only provides a basic framework of the overall assembling of the vertical circuit. There are many more connectivity has to map on top of this framework. For example, interneuron has to recruit to individual functional clonomic circuit. And recently, we also showed that a long-range connection, for example, between the frontal cortex or sensory cortex, they can also be influenced by this type of lineage-dependent microcircuit to microcircuit communication. So I'd like to spend the last few minutes talk about something unpublished. We know that besides the functional column, the neocortex also have another important organizational feature, that is a functional map. We call it neural map. For example, this is a pioneer work done by Clay Reid's group by using 2-photon imaging to look at the orientation tuning synactivity in the layer 2-3 of the left visual cortex. So each color here represents that neuron will be activated or tuned to a selective orientation that moving across the visual field. So for example, they found in a rodent or in rat, the map of the orientation selectivity should exist as a soft pepper pattern, which means even neighboring two neurons can tune to a very different orientation of light stimulation. And this is quite different from the clustered map, neural map we often see in higher mammals such as cat, primate. For example, neurons tuned to similar orientation will be clustered together. They can have transition from one cluster to another cluster, or even have pinwheel structure, but overall you have this clustered map. So why would we have these two different type of map, the salted pepper pattern map or clustered map? And this raised a fundamental question in terms of origin and significance of the neural map. So we like to test whether those framework of the circuit assembling in the neocortex, depending on the early process of neurogenesis and neuronal migration, can influence or contribute to those map formation or the salt pepper map formation in the mouse neocortex. So we come back to this early events of neurogenesis and neuronal migration. We can see that they make the switch from proliferative division to neurogenic division around E11 to E12. So namely, if you enable a clone during the symmetric proliferative phase, you will see a much larger clone. And within this clone, you have both sister and cousin. We call those sisters are the neurons coming from same asymmetric dividing neurogenic radioglial progenitor cells. But if the neurons coming from two symmetrically, initially symmetric dividing radioglial progenitor cell, we call between those two individual clones that we call them cousins. So in other words, at E11, enable symmetric dividing radioglial progenitor produce clone, you have both sister and cousins. But if you enable the clone at E13, that you will only have asymmetrically neurogenic radioglial progenitor cells. They only produce a sister clone. And we found that the sister and cousin neurons actually exhibit distinct synaptic connectivity and functional similarity. In other words, the sister cells is preferentially connected and show strong functional similarity, but actually cousin neurons do not show preferential synaptic connectivity. In fact, horizontally distributed cousin cells actually will antagonize synaptic connection and also show functional diversity. In other words, they will show much more diverse functional tuning properties compared to control neurons. In fact, you can simulate the functional map based on this lineage relationship and their functional similarity. For example, we find that in the superficial layer of the mouse neocortex, each individual radioglial progenitor produces about 5 neurons. So if we simulate 25 neurons based on the 5 neurons will be the sisters, we can get 20% of them are sisters, 80% of the cousins, and based on sister and cousin neurons tuning similarity, we actually can simulate the neural map, which shows this unit sort of pepper pattern, similar to random, indicating this lineage-related functional similarity and synaptic connectivity can nicely predict the neural map organization, in this case, in sort of pepper pattern. Moreover, if we increase the number of neuronal output, in other words, instead of produce 5 neurons in layer 2 to 4, they'll produce 10, 15, or 20, or 25. As you can see, the overall ratio between sister and cousin progressively increase. And this because of the sister neurons share functional similarity and cousin neurons actually can have a distinct functional tuning properties. And this can gradually shift a sort of pepper pattern neural map to a more clustered neural map, indicating that even we have 2 different types of map, but this map may have a similar mechanism just depending on the output or number of neurons. Of course, this is a hypothesis. We like to test this in the future. So the idea is that even those 2 maps may look different, but they may have similar developmental mechanism. The key difference is that the neurogenic unit size difference. In a salt pepper pattern map, the output of individual radioglial progenitor cell in terms of number of neurons produced is relatively small, but in a clustered map, the output of individual neurogenic radioglial progenitor cell will produce a much larger size of neuronal output. And this is actually consistent with the current idea. We know that during the cortical expansion, because of the intermediate progenitor cell or transient amplified progenitor cell, the output of individual radioglial progenitor cell in terms of number of neurons is expanded in the high mammals, for example, in the primate, in ferret or cat. So we like to test this idea in the future. So to summarize this part, we think there is this lineage-dependent progressive assembling of the neocortex from the neurogenesis neuronal migration to gap junction, chemical synapse formation to the clonic circuit to even neural map assembly. And this will provide a basic process for the functional organization, the functional assembling of the neocortex in a stepwise manner. So I'd like to finish here by thanking the people who actually did the work. As I mentioned, that the Gliogenesis work was largely done by 2 talented students, Zhongfu Shen and Yang Ling, and also the last part of the lineage-dependent functional assembling, the electrophysiology, I didn't have time, done by Xinjun Zhang, a previous postdoc in the lab, and also the functional map tuning map analysis was done by a talented student, Yang Ling, in the group, with close collaboration with Ben Simons and also Kenichi Oki. Those are the collaborators that share the reagent with us. Those are all funding sources. Thank you very much for your attention. Happy to take any question.
Thank you very much, Songhai, for a great talk. Not surprisingly. There are a number of questions that I'm going to ask you, and you can then respond to them. And if you need to go back to your slides, you can always share the screen again, etc. So the first question is from Isabel Martinez Garay, who is asking, what would determine the timing of the individual divisions of each radial glial cell? Cell cycle length is known to increase over development, but that was not necessarily reflected in either of the 2 models that are shown. So
yeah, this is a great question. You know, of course, as in the question that the cell cycle length overall, I think, is lengthening as development proceeds. we like to think that there's an intrinsic clock of the radioglial progenitor cells. In other words, for example, when I say that during the proliferative division, at each round of division, it will lose the proliferative potential, which means the radioglial progenitors almost can't count the round of division it goes through. Of course, we don't know what is the molecular control of this process, and what is drive in terms of if you have divide, for example, during the early phase of neurogenesis, then you have to influence the late phase of neurogenesis, which, as I showed, there is this inverse correlation between superficial and deep layer neuron production. And so a short answer is I don't know what controls the division, but I think with this the powerful single cell type of analysis, I think we may be able to find out the precise molecular regulation in the near future. For example, we think that there are some, for example, molecules that progressively increase as radioglial progenitor cell progress through the lineage from proliferation to neurogenesis to gliogenesis. In fact, some of the candidate molecules, if we manipulate the expression level, they actually can change the round of division, which means they can make the clone bigger. So clearly suggesting that this cell cycle, round of the cell cycle, and also the time of the division can be regulated by molecular mechanisms. So I think they're likely to be both intrinsic and also extrinsic type of regulation that controls the overall process of the radioglial progenitor cell, both as a population and also as at the individual radioglial progenitor cell level.
So if I can follow up on that, there's some work from Jürgen Knoblich, and especially in Drosophila, neural stem cells that are neuroprogenitors called neuroblasts that are dividing, that suggest that they get smaller as they divide, right? Their size gets smaller, and so there's quite nice work, both direct and indirect, and from Jürgen and others, that shows that there's a coupling between the metabolic state of the progenitor cell and then the mediator complex. And gene expression and so on, and also proteins that regulate translation like BRAT, which I believe is called TRIM in vertebrates. Those increase in level as the progenitor divides, and so they repress protein translation and so on. Suggesting that basically this reduced metabolic capacity makes the cell smaller and makes future divisions sort of less likely to be proliferative. Is there any evidence in the mammalian literature around this concept?
Yeah, I think that what you referred are both, I think, relevant to the mammalian system, even may not be published yet. For example, the metabolism, I remember Jürgen's, like this oxidative stress actually can kick the progenitor cell out of the cell cycle. In other words, why do radioglial progenitors exit the cell cycle at the later stage? So clearly there is this metabolic influence. For example, the oxidative stress, I think in the cortex also. For example, during the early phase, proliferation to the neurogenesis to the gliogenesis is very likely influenced by the overall metabolic regulation. And also, as you referred, that Chu Ming Li showed that there's this gradient, both molecule of the upward gradient and downward gradient that can control the fly lineage. So I think the similar mechanism can be seen in the cortex.
Yeah, I mean, there's definitely evidence that different types of stress, either due to viral infections or unfolded protein response or ER stress, etc., can alter the proliferative versus neurogenic divisions of cortical progenitors. So, okay. So the second question is from Santos Franco, who is asking, RGPs, I guess radioglial progenitors, that are relatively quiescent. That's a really good question. So radioglia that are quiescent when you're doing the madam labeling are likely to be missed, or that are cycling very, very slowly or whatever. So do you have a sense of what proportion of radial glia at any given time point are actually difficult to label by MADAM because they're not dividing or whatever, and so likely to be missed. And whether those represent a peculiar, that might explain the discrepancy between the two models in a way, I guess.
Yeah, this is a great question. So clearly, the MADAM relies on the cell division. So if cell doesn't divide, of course, they will not be labeled. So what I can say is that over the past 5-6 years, with different type of the Cree lines, we have labeled on the order of 1,000 or more clones. Those clones appear to be quite consistent to each other, but we cannot rule out there are some small proportion of radial glial cells are not actively dividing that we are not sampling them. And as I said, we use nesting Cree, we use EMX1 Cree, and also Hefner's group use SOX2 Cree. So clearly this is not something to do with the Cree,
right.
Different type of Cree.
Right.
And then, so whether there are some quiescent radial glial progenitor cells that will, for example, you label at the early stage, then turn on to divide to produce late-born superficial layer neurons. I think it could be. There might be the difference. You use GFP reporter, as in Oscar's group did. But the one thing is that appears those clones appears to be small, and on the order of 2 to 4 cells, and also they different from the majority of the clone, which means they're often to be either superficial or deep layers. So whether those represent those quiescent clones, so if those are the quiescent RGPs, then they will be different. But as I mentioned, that there is a debate whether actually those reflect some of those unusual intermediate progenitor cells, because there are intermediate progenitor cells divide more than one round.
That's
so
right.
if they represent intermediate progenitor, then you will expect they will be smaller, they will be localized to fewer layers.
Okay. So I guess the mirror image of that would be cell death, right? And this is a great question by Fernando Garcia Moreno. I like this question a lot as well. So to what extent could apoptosis of neurons after they've been produced really, in a way, sculpt the size of the clone and its distribution so that it looks like you have this sort of, highly reproducible, more or less within boundaries, of course, neural progeny, and to what extent could therefore this kind of apoptosis also direct your interpretation of transitional clones, right?
Yeah, so in fact, we have been thinking about this cell death quite a bit. So with the MADAM, you actually can have some sense of cell death, because that's the reason we focus on green-red clones. So sometimes we see a single green or single red clone, which you clearly know the other half of the clone is lost in the process. So we eliminate those single color clones, so just to be sure that we look at the full clone. So this is one. In terms of the Neogenesis, what we did are 2 things. So first of all, we look at the overall single colored G2X clone versus double colored. That is only about less than 10%. So indicating that at least the embryonic death is not a majority of the process. Then another experiment, what we did is that we compared the clone labeled at P7 versus P30, right? Because Neogenesis is largely complete by at the birth time. And if you look at if there's quite a bit of cell death and neuronal death during the postnatal development, then you would see a clear size difference between the clone size at P7 versus P30. So we cannot fully rule out there's any significant neuronal death, but or as estimation of the contribution of the death, at least for Neogenesis, is not that extensive. In other words, we didn't observe a drastic clone size change between P7 and P30, for example. But I mean, the ultimate proof would be to suppress the cell death.
Yeah.
To see whether the clone size look different or distribution.
I think that's because it could because the death could also occur much earlier. So during immediately post division, right?
Yeah. In fact, there's this interesting poster I saw in zebrafish. Actually, there can be entire clone die.
Yeah.
So that would be also an interesting phenomenon, but that would not affect the overall. Final analysis of the clone.
No, if the entire clone does. Okay, so we have 4 more questions. I hope you still have time to stay with us.
Sure.
Again, another question from Isabel is: does the type of clone for astrocytes and oligodendrocytes change with time, as it is with neurons?
Type.
I guess does the proportion of O versus O and A clones shift over time? I think.
Yeah. So this is a good question. So we only look at the P30. So we varied the labeling time. For example, we label at 13, 14, 15, 16, but the final examination were done at the P30. So at P30, that's what we find about 25, 60, 25, 15% of this ratio. But the question is that if we look at this ratio at the early time point or a later time, we don't know.
No, I guess if you look at the ratio of clones produced at, say, E13 versus E16, is the ratio different?
This is relatively consistent.
Okay.
So the labeling time, for example, 13, 14, 15, we know that this ratio is largely comparable.
Okay. I think that was the question, but I'm not sure. Okay.
Yeah.
So so then we have a question from Jorge Garcia Marquez, who's asking, in the NF1 mutant, have you analyzed older brains, so 2 to 3 months? In the normal brain, NG2 glia clones keep growing to reach up to hundreds of cells in a few months. I wonder if there's a limit in the number of NG cells per clone.
Yeah, this is a very interesting question. So we have looked at them, a few of the brains to more than 2 to 3 months. So in some of those brains, they have this huge clones of the OPC cells with hundreds to thousands of cells. So part of the reason is that we want to see whether we actually can create a tumor from a single cell mutation that we wrote. But this has been done a bit, for example, by Hui Zhong. In a single P53 mutant, actually you can rarely create a tumor.
Yeah.
So you need another mutation like P53 with NF1 that you may be able to generate the tumor. So in that regard, we are still trying to figure out whether we actually aging the animal, we actually can create the tumor.
Yeah.
But I like to say that we think that those clones are not like, of course, we don't have the evidence. So you could argue that those huge clone can come from embryonic gliogenesis or come from progressive, because OPC can proliferate even in the adult brain.
Sure.
So we like to think that because we observe the large clone even at the early stage, so we likely to think that those large clone we see is not because of cumulative OPC proliferation, but rather it's because of the intermediate oligodendrocyte precursor cells. They have this unusually large output, but we will have to test this directly.
Okay. So then a question from Michael Ratz, who's asking, how do sister cells that form long-range connections actually know that they belong to the same clone? So what's the,
this is this is a great question. That so what we find is that if we use rabies virus to label a clone in the S1 or the V1, what we will find is that the presynaptic cells in, let's say, in M2 or singular cortex in the frontal area, they often also exist as vertical clusters. And even you record from those vertical presynaptic cells, they have a higher connectivity. So this indicates there is a microcircuit in the S1 communicate with the microcircuit in the frontal area, in the M2 or cingulate cortex. So how does this happen? We don't really know. We only can speculate. So one possibility is that the input of the sensory cortex, like the bottom-up communication, is not random. So in other words, there's a possibility that the bottom-up input from the sensory cortex to the frontal area is already specific. And this may help to drive this type of the local circuit formation.
But sorry, go ahead.
Another possibility is that this type of vertical structure of the neurogenesis neuronal migration occurs in all the area of the cortex. So in other words, even in the frontal area, you will have this local vertical clusters. They will have this gap junction likely, and they serve as a building block for the local microcircuit. And what then the sensory input, top down, bottom up, will just couple them together. And this is another possibility, but we don't know. We haven't demonstrated that is a sister clone in S1 and sister clone in M2.
Right.
We will not be able to do this, but we demonstrate that a sister clone in M2 can communicate a vertical cluster in the S1, or a sister clone in S1 can communicate with a vertical cluster.
So if I can follow up on that, I don't know whether this makes sense, but if the vertical clusters that have a higher probability of connecting to each other are actually born more or less at the same time, which means that they will extend their axons and so on at the same time, then that gives them an advantage. So if I'm, if I'm going to explore a certain area, right, I'm more likely to connect to neurons that have already reached a certain level of maturity than to ones that are still migrating, obviously, right? So if there's some sort of, and there you don't necessarily need coupling, you just by the accident, quote unquote, of the timing of maturation, your most likely partner in the area that you're innervating is the one that has matured at more or less the same time as you. So you get it through time, you get a restriction of possibilities. Is that, I don't know enough about how these circuits develop, but is that
so in fact, this actually is something demonstrated by I forgot his name, in Switzerland, in the hippocampus. For example, the CS3, CA1 connections that can be regulated by the burst timing of neurons. So this long-range connection. But in the cortex, so we are trying to explore this because to monitor the activity and to monitor the axonal projection is because the distance is quite long.
Of course. Okay, so the last question, and I'm sorry it took us so long to get to
no problem.
From Yasushi Nakagawa. Yasushi is asking, very nice talk, Songhai. Do you observe more tangential dispersion of glia than of neurons in a given madam clone? So a madam clone that contains both, do the glia disperse tangentially more than the neurons, I guess?
Yeah, I think in general, probably I would say yes, but we haven't quantitatively compared them because the glial cell produced after neurogenesis. So in a sense that, especially in the white matter, there will be a bit more natural dispersion because in the later stage, the brain already grew quite a bit. So the original position of the radioglial cells, as they lose their ventricular infit began to produce glia, they have more range in a sense to, and also because they can deposit the local precursor cells, and that produce probably a bit more natural dispersion. But they overall stay in a cluster, so you actually can see them. I couldn't show you the movie because I had to switch the computer. So you can, with a sparse labeling, you actually can see them quite close to their sibling neurons.
Okay. Well, that's all the questions that we had. Thank you very much for your patience.
Sure.
Thank you everyone who stayed with us, also for your patience. Hopefully the vaccines are coming soon, and we will be liberated from this pandemic, and we get to see each other again. And in the meantime, please stay safe, take care of each other and yourselves. Thank you again, Songhai.
Thank you very much, Nathan.
Take care.
You all. Bye.
Bye.